So hello guys, today we'll discuss the immune complex mediated type 3 hypersensitivity. So we discussed already type 1 and type 2, and what is the type 3 reaction. Also today we'll discuss the type 4 reaction, and also we'll discuss a few autoinflammatory autoimmunity pathologies, autoimmunity diseases, and their basics of their appearance. It's only basic. I want to say to you, only just basics, because in detailed way. So you'll discuss it in clinical subjects. Okay, antigen antibody complexes produce tissue damage mainly by eliciting inflammation in the sites of deposition. So you remember that antigen antibody, when they attach together, they promote the activity of complement system. Okay, let's check it out. So you see, we have free antigens which are present in circulation, and antibodies, and free antibodies which are produced by plasma cells. So in which cases it might happen? For example, some bacterial invasion. So in some bacterial invasion, so when antigens present in that circulation, or might be by ingestion of some compounds inside the and somehow. So anyway, so we have different pathways of this activation, but somehow antigens are present inside the bloodstream. And when it's present in bloodstream, these antibodies start to attach with these antigens, and create the antigen antibody complexes, large antigen antibody complexes. Let's talk about this. They're like immune complex mediated diseases tend to be systemic, but often preferentially involve the kidney. Glomerulonephritis is the most common disease. Sometimes joints, they cause arthritis, and small blood vessels, they cause vasculitis. All of which are common sites of immune complex deposition for reasons mentioned below. So here we'll discuss these reasons later. The hype. So okay, I won't join no one else. Okay, systemic immune complex disease. So it's also sometimes known as serum sickness. It's a prototype of systemic immune complex disease. It was once a frequent sequela of administration of large amounts of foreign serum. So in which case we have a lot of foreign serum. So in a transplantation, so I mean hemotransfusion, for example, or might be serum of so serum. We use serum for passive immunization. Yeah. So and that's why that's example here we have it's a serum from immunized horses used for protection against diphtheria. So we use this passive immunization by injection of antibodies inside our body. But sometimes these antibodies became antigenic for us, and our own cells, our own immunity cells, produce the antibodies against these antibodies against antibodies, and that's serum sickness is a typical example. Okay, let's watch it. And it has three phases. This disease has three phases: the formation of immune complexes. So introduction of a protein antigen triggers an immune response that results in the formation of antibodies, typically about 1 week after the injection of the protein. These antibodies are secreted into the blood, where they react with an antigen still present in the circulation and form antigen antibody complexes. So you see, so as you remember, antibodies are not secreted directly after as a first response. It is produced like second response in adaptive immunity. So and then when it enters into the body, first time enters into the body, so we don't have immunity against it. It happens in secondary, second time, when second time you inject this serum, and second time you inject this antibody against which you have your own antibody. Okay, deposition of immune complexes. So how deposition of immune complexes happened? In the next phase, the circulating antigen antibody complexes are deposited in vessels. The factors that determine whether immune complexes formation will lead to tissue deposition and disease are not fully understood, but the major influences seem to be characteristics of the complexes and local vascular alterations. So you see, these antigen antibody complexes with no proper understanding how it happens, they are deposed in blood vessels. Especially in which blood vessels? So peripheral blood vessels with most tiny blood vessels. Most tiny blood vessels, so which are present in glomerular tissue, and also in joints. Also, it present, and also in all small vessels, which cause the vasculitis. Okay, let's so organs where blood is filtered at high pressure to form other fluids like urine and synovial fluid are sites where immune complexes become concentrated and tend to deposit. Hence, immune complex disease often affects glomeruli and joints. So you see, deposition of immune complexes might be present everywhere, but predominantly when we have higher pressure, when high pressure is present. This is synovial fluid and kidneys usually. That's why some autoimmune diseases are affecting first of all kidneys and joints. Okay, and third one is inflammation and tissue injury. So immune complexes. So you remember that when immune complexes are antigen antibody complexes, they activate the same complement system. But you see, blood vessels are not primarily antigenic for us. But after the deposition of antigen antibody complexes, it starts to be antigenic. It starts to work as antigen, like antigen antibody reactions happen here, and then causes the inflammation, and then causes a tissue injury by first of all by complement system, but secondary with immune cells activation by like macrophages and leukocytes and even lymphocytes activation. So this is third type of immune reaction. Okay. In fact, serum C3 levels can, in some cases, be used to monitor disease activity. So, when in this decreased C3 levels, decrease of C3 levels means that C3 splits out, and antigen antibody complexes are produced, and that's why we can see that C3 levels in the body are decreased. Usually, very rarely it's used in clinical practice, but in this disease we have to check it out. Okay, and might be remember from second chapter we discussed about fibrinoid necrosis. Yeah, so okay, let's take a look on this diagram. So this is the first reaction. Then antibodies are attached with antigens, and then antibodies with antigens are attached to blood vessel somehow, and then it causes the activation of neutrophils and macrophages and complement, and it causes destruction of blood vessel here and formation of immune responses like vasculitis. So here in connective tissue of blood vessel, around the blood vessel, we have a inflammatory reaction, and typical, acute, and then with attempts to chronic inflammatory reaction, because we can't eliminate the offending agent because we have no offending agent. Yeah, we have only our own immunity reaction. And that's why it starts like acute inflammatory reaction and then continues like chronic inflammatory reaction. As that happens, vasculitis, and this vasculitis forms a special type of necrosis known as fibrinoid necrosis. Yeah, because it starts to kill our own cells, and this vasculitis surround our own cells, and debris of the cells promote our own immunity against this debris of cells. And then you see it's like cascade of reactions which are finally causing the fibrinoid necrosis, this tissue. Okay. So inflammation tissue. Okay, morphologically, it looks like fibrinoid necrosis. We discussed this fibrinoid necrosis. It's intense neutrophilic infiltration because of acute reaction. Necrotic tissue and deposits of immune complexes, complement, and plasma protein appear as a smudgy eosinophilic area of tissue destruction, and appearance termed as fibrinoid necrosis. Okay, and we have local immune complex disease. This local immune complex disease is, for example, it's Artus reaction. It's localized in the area of tissue necrosis resulting from acute immune complex vasculitis, usually elicited in the skin. The reaction can be produced experimentally by intracutaneous injection of antigen in previously immunized animals that contain circulating antibodies against the antigen. Okay, example. So you know this. In Kyrgyzstan, I don't know how it happens in Pakistan, but in Kyrgyzstan, as I know, we have a Mantu reaction. Mantu reaction. So Mantu reaction is so by this we check our immunity against tuberculosis, bacteria tuberculosis, and this bacteria tuberculosis. So sorry. bacteria tuberculosis, tuberculosis bacteria. So it causes our immune response, and almost every one of us possesses this immunity against the tuberculosis bacteria. And we have to check the state of our body. For example, we just inject a little bit of antigen of this bacteria. Um, and we check the immune reaction on the body. So we check how much is it how much is hyperemia here, how many blood vessels are produced as attempted healing of this during this injury. Okay, antibodies and immune complexes may deposit in tissues or blood vessels and elicit an acute inflammatory reaction by activating complement with the release of breakdown products by engaging Fc receptors of leukocytes. The inflammatory reaction causes tissue injury, as we discussed before. Antibodies can bind to cell surface receptors or other essential molecules and cause functional derangements, inhibition or unregulation activation without cell injury. Okay, okay, this example, one of examples of this we'll discuss later. So, what is it? Deposition of antibodies without injury. Okay, but now let's switch to another type 4 hypersensitivity. Type 4 hypersensitivity is directly, it's known as T cell mediated. T cell mediated. So it means we use T cells, and of course you can suggest it's T cells. And we have two types of T cells: CD4 positive and CD8 positive, and both of them both of them are used in this hypersensitivity. Both of them are active in this hypersensitivity reaction. Okay, for example, CD4 positive T cells mediate hypersensitivity induced by environmental and self-antigens is the cause of many autoimmune and other chronic inflammatory disease. Cell killing by CD8 positive cells, cytotoxic T cells, may also be involved in some autoimmune disease and may be the dominant mechanism of tissue injury certain reactions, especially those that follow viral infections. Okay, let's talk about CD4 positive T cell mediated inflammation. CD4 positive T cell mediated hypersensitivity reactions, cytokines produced by T cells induce inflammation that may be chronic and destructive. So remember, CD4 positive cells, they don't kill directly cells, but they cause the formation of a lot of cytokines and affect more and more inflammatory cells to this area. And this T cell mediated inflammatory reaction is sometimes in some books you can find this name, delayed type hypersensitivity. Because all other types we discussed before, they appear directly after the injection of these antigens into the body. But this one, so you see, it's appeared much slower, much lower, much more long activation of these cells, because it's CD4 positive T cells. It requires activation of once on this side and then switch to another side and then activate later. That's why this reaction happens in 24 to 48 hours. That's why it's named as delayed type of hypersensitivity. So it's shown here, a contrast to immediate hypersensitivity. So both cells, Th1 and Th17 cells, we discussed it before, but no need to remember them. They're involved. They're involved in this immunity reaction. Okay, let's talk about how activation of CD4 positive T cells happen. So I remind you, CD4 positive cells are activized predominantly by the dendritic cells, because dendritic cells possess MHC2 and this so they are displayed by MHC2 by dendritic cells, and after the naive CD4 positive T cells, they read the information from dendritic cell, and due to this, dendritic cells produce interleukin 2. And then it causes the autocrine growth factor to stimulate proliferation of antigen-responsive T cells. So you see, CD4 positive T cells, after activation by dendritic cells, they cause its own proliferation. It's autocrine growth factor. It means our own cells are producing hormone to activize itself. So, but it happens only after the activation with the dendritic cell. Okay, subsequent differentiation of antigen-stimulated T cells to Th1 or Th17 cells is driven by the cytokines produced by APCs and at the time of T cell activation. So the same process happens to differentiation, but don't care about this. In some situations, the APC produces interleukin 12, which induces differentiation of CD4 positive T cells to the Th1 subset. Interferon gamma produced by these effector cells promotes further Th1 development, so this subpopulation of T lymphocytes, which also amplifies this reaction. But don't forget that interferon gamma, another activity of interferon gamma is activation of macrophages, so just keep it in mind. If the APCs produce the inflammatory cytokines interleukin-1, interleukin-6, and a close relative of interleukin-12 called interleukin-23, so that's no need to remember, T cells are induced to differentiate to the Th17 subset. So you see, some cytokines reaction so causes the differentiation of CD4 positive T cells into the Th1 or Th17. So that's depend, and the reaction depends on it. Okay, so responses of different differentiated effector cells. So you see, Th1 cells secrete cytokines, mainly interferon gamma, which are responsible for many of the manifestations of delayed type hypersensitivity. So you remember, I already mentioned you that interferon gamma activated macrophages by classical pathway are altered in several ways. Their ability to phagocytose and kill microorganisms is markedly augmented. They express more class 2 MHC molecules on the surface, enhancing antigen presentation. They secrete TNF, interleukin-1, and chemokines, which promote inflammation. So that's the role of macrophages we discussed before in chapter number 3. So that's why I just repeat it to you. So, but you have to explain it to me in practical class. They produce more interleukin 12, amplifying the Th1 response. Okay, let's skip it. Let's skip it. So it's not so important. Okay, clinical examples. So which clinical examples of this we know? The classic example of DTH is a tuberculin reaction, which is produced by the intracutaneous injection of purified protein derivative. So we have purified protein derivative, so which is so also reaction of immunity reaction against our tuberculin. So it's protein containing antigen of the tubercle bacillus. So and then we wait for 24 to 72 hours. And after this, we check how much it present, how severe it is. So and if it's severe present, so it must be our immunity is very tangent with bacteria tuberculosis. So might be we have very active reaction against the tuberculosis right now. But if it's a very slow reaction, very little reaction, it's present like hyperemia of skin. Yeah, if hyperemia of skin is present, so if it's normal size, it means yeah we have immunity, but it's not active immunity. But if it's too large, we have active immunity. It might be right now we have some tuberculosis in our body. So tuberculosis is active in our body, might be. But if it's too little, so it means we don't have immunity against it. And if your body will interact with the bacteria of tuberculosis, so might be you will suffer with tuberculosis later. Okay. Then, so 1 minute. Let's check. So. So, for example, we have another disease which is known as contact dermatitis. Contact dermatitis is a common example of tissue injury resulting from DTH reaction, delayed. Delayed type of hypersensitivity, and usually it's sometimes it may be evoked by contact with urushiol as antigenic component of poison or ivy or poison oak, and present as an itchy vesicular blistering dermatitis. So okay, just contact dermatitis is an example of this. and CD8 positive T cell mediated cytotoxicity. So in this is most dangerous. The most dangerous type of cytotoxicity, because cells are directly kill antigen expressing target cell. This tissue destruction by CTLs may be a component of some T cell mediated diseases such as type 1 diabetes. So type 1 diabetes, I want to remind you that, as we will discuss it later in diabetes disease, but T cells in some types of type 1 diabetes, so our own immunity T cells start to kill Langerhans cells of the pancreas. And it causes the first type of diabetes, so insulin insufficiency type of diabetes. Okay, and also in the virus-infected cells, viral peptides are displayed by class 1 MHC molecules, and the complex is recognized by the TCR T cell receptors of CD8 positive T lymphocytes, and it causes the killing of infected cells. But sometimes it goes too fast, and as we discussed so before with the group number 4, that T lymphocytes, which are T lymphocytes are very active in viral infection and might be too active. And here in this book, it's shown as an example is viral hepatitis. But I want to explain another processes like um, uh, coronavirus. Of course, coronavirus. So because coronavirus also causes same defect in the lungs. So coronavirus affects the lung tissue, so alveolar cells of the lung, and they destroy this alveoli of lung because they are infected with virus. And of course, it kills us faster than virus itself. So our own immunity reaction, so hypersensitivity reaction, kills us faster than itself. Okay, principal mechanism of T cell mediated killing of targets involves perforins and granzymes. I remind you that in T cells, so in cytotoxic T cells, we have perforins and granzymes, and main function of them is perforation of cell membrane of our own cells or bacterial cells or any antigen bind tissue. Um, the secrete complex of consisting to perforin, enzymes, and other proteins that enters target cells by endocytosis. In the target cell's cytoplasm, perforin facilitates the release of granzymes from the complex. Okay, granzymes are proteases that cleave and activate caspases, which induce apoptosis and the target cells. So you remember you remember in chapter number 2 you said, yeah, so how immunity causing the apoptosis. So that's why. So granzymes directly cause activation of caspases without its extrinsic pathway of apoptosis, if you remember. So activated CTLs also express Fas ligand, a molecule with homology to TNF, tumor necrosis factor. It also triggers the apoptosis because we have TNF receptors on every cell, and we have Fas receptor expressed on target cells, and that's why we have apoptosis as response of cytotoxic T cells. CD8 positive T cells also produce cytokines, as we discussed in CD4 positive interferon gamma, and also it causes the affection of other immune cells like macrophages to the site of infection, to the site of viral infection, and it also take part in a hypersensitivity reaction. So you see the border between hypersensitivity and normal sensitivity is shadowed. Yeah, so actually, so normal response to virus so can be dumped with abnormal reaction. So to acute reaction to the same stimuli. So that's idea. Let's talk about autoimmune diseases. We have a lot of autoimmune diseases, so here you can read about this. It's quite interesting, but it's I don't want to spend our time to this. So first of all, let's talk about immunologic tolerance. Our own cells, they have some specific immunologic tolerance. So what is this phenomenon? It's a self-tolerance. You remember that every cell of our body expresses the MHC ligand, MHC1. Yeah, MHC1 is the main protein which facilitates the self-tolerance. So if we have in our body, use it like a passport. Yeah, and if we have Okay, we have okay. Let's talk about it step by step. We have self-tolerance. Self-tolerance is subdivided into central tolerance and peripheral tolerance, and which let's discuss about it. So, what is a central tolerance? So you see, immature self-reactive T and B lymphocytes, clones that recognize self-antigens during their maturation in the central lymphoid organs, are killed or rendered harmless. So you know that you remember that we have lymphoid organs, and then it's a sites of reproduction of lymphocytes. And naive T cells, they have no charge to kill cells. And B lymphocytes, which are not affected in production of antibodies, they are not they don't act as killing cell. But in immunity organs, we have dendritic cells which present this antigen on the surface, and then B lymphocytes and T lymphocytes, they start to be activized by these antigens, and they start to produce. So, in case of T lymphocyte, T cell receptors (TCR). In case of B lymphocytes, they start to produce antibodies. But you know that sometimes, so our body understands that sometimes so antibodies we produce and T cell receptors we produce, and so they can be injurious to our body. And that's why if we recognize that MHC is killed by this thing, by this reaction, so we have to, if our own protein is killed by this, so we need to depress their activity. So how it happens? Yeah, we have some thoughts about it, but it's very complicated process, so it's cannot be explained in one point. So let's discuss it in basic pattern. When immature T cells expressing TCR, T cell receptor specific for self antigens, encounter these antigens in the thymus, signals are produced that result in killing of the cells by apoptosis. So you see, we have this process known as negative selection. These T cell receptors, which are expressed against our own antigens, they are present in thymus, and that's why we have negative selection. So when thymus cells, they understand that this T lymphocyte gonna kill our own cells, they cause a negative selection, or it's also known as clonal deletion. So we delete these clones of cells to prevent their activation in future. Okay, then a wide variety of autologous protein antigens, including antigens thought to be restricted to peripheral tissues, are processed and presented by thymic antigen-presenting cells in association with self-MHC molecules, and can therefore be recognized by potentially self-reactive T lymphocytes. A protein called AIRE, autoimmune regulator, so stimulates expression of some peripheral tissue restricted self antigens in the thymus, and is thus critical for deletion of mature T cells specific for the antigens. So okay, let's say about one example. We have autoimmune polyendocrine syndrome. So it happens when we have mutation in this ARE gene. So this, in case of mutation in ARE gene, so when suppression of this cells, so as I said, negative selection is dumped. So of course we have, as example, the autoimmune polyendocrine syndrome, and this autoimmune polyendocrine syndrome causes endocrinopathies in a lot of organs. Okay, let's discuss it in chapter number 24. Yeah, so it's much later we'll discuss it. In the CD4 positive T cell lineage, some of the cells that see self antigens in the thymus do not die but develop into regulatory T cells. Okay, let's continue. When developing B cells strongly recognize self-antigens in the bone marrow, many of the cells reactivate the machinery of antigen receptor gene rearrangement and begin to express new antigen receptors not specific for self-antigens. This process is called receptor editing. It is estimated that 1/4 of the to 1/2 of the B cells in the body may have undergone receptor editing during their maturation. So 1,4 only. They change their receptors, and that receptor deletion does not occur. The self-reactive cells undergo apoptosis, thus purging potentially dangerous lymphocytes from the mature pool. So that's okay. I hope you understood. So central tolerance, however, is imperfect, and not all self-antigens may be present in the thymus and bone marrow, and hence lymphocytes-bearing receptors for such autoantigens escape into the periphery. So, and that's why all the time we have some kind of autoimmune reaction. Self-reactive lymphocytes that escape negative selection can inflict tissue injury unless they are deleted or muzzled in the peripheral tissues. Okay, let's talk about peripheral tolerance. So what is a peripheral tolerance and how is it maintained? So we have a few mechanisms. For example, anergy. Anergy is when lymphocytes that recognize self-antigens may be rendered functionally unresponsive, a phenomenon called anergy. We have activation of antigen-specific T cells requires two signals: recognition of peptide antigen association with self-MHC molecules, you remember, yeah, which are present on the surface of APCs, and a set of costimulatory signals, like second signals. First of all, we discussed like CD28 signals, yeah, and other costimulate like B71, B72 on IPCs. Okay, anyway, so it's a deep down question. So, but you remember that it's activized. If the antigen is presented to T cells without adequate levels of costimulators, the cells become anergic. So you see, if we just switch off, so we have this receptor. So that's why we have so complex inflammatory reaction. Yeah, we have so complex immune reaction because along with the T cell receptors, we have also CD28. So and sometimes we can just turn it off. Turn it off and or maybe turn off B7, B71 or B72, and that's why in peripheral organs when already charged T lymphocyte enters to the site, but it doesn't it causes an anergy. Okay. Energy also affects mature B cells in peripheral tissues. It is believed that if B cells encounter self-antigen in peripheral tissues, especially in the absence of specific helper T cells, the B cells become unable to respond to a subsequent antigenic stimulation and may be excluded from lymphoid follicles, resulting in their death. Okay, and then another effect, we have suppression by regulatory T cells. So you remember in the beginning we discussed that we have T helper cells which are predominantly responsible for regulation of this. So I don't want to explain you all this stuff. That's T cells, high level CD25, the alpha chains, interleukin 2. So just skip it. But we have suppressor cells, T suppressors, which suppress the activity of some lymphocytes peripherally, not only T lymphocytes but also B lymphocytes, and both of them can be controlled by them. So how it happens? So just remember, it's somehow CD25 genes or transforming growth factor. So okay, uh, so no need to remember. It's so actually we don't have so much time to discuss it. And deletion by apoptosis. So you see that when cells are recognize self-antigens, they can receive signals that promote their death by apoptosis. So similar like happened in central tolerance. So how apoptosis happens? So you already remember. Okay. So then, and some antigens are hidden, sequestered from the immune system because the tissues in which these antigens are located do not communicate with the blood and lymph. So, for example, we have immune privileged sites. Yeah, and examples are examples of this. So just imagine. So during the formation of self-tolerance, they don't interact with these antigens. And in central tolerance, we don't have specific. So we don't have this marker that this is self-antigen. But in case of injury of this antigen, injury of this structure, which Okay, example. Yeah, so I'm urologist. You know that I'm urologist, and I give an example from urology. Testis, yeah, testis possess a blood testis barrier. Blood testis barrier, because inside the testis are produced cells, are how it's called, um, by they produce cells by meiosis, and due to meiosis, produce cells with different antigen composition. So you remember with their so they're one end composition cells. So haploid cells, yeah, and normal cells are diploid, and haploid cells, which are present inside the testes, now they are antigenic for us. But our cells, like Sertoli cells in testes, yeah, so maybe you remember it. They prevent the antibody entrance to the site. But in case of injury of testes, so I mean mechanical injury, so it seems like this barrier is dumped, destroyed, and then our immunity cells can easily penetrate there, and dendritic cells penetrate there and found these antigenic cells and start to kill them, and starts to send the signal to kill them. Yeah, you remember dendritic cells send the information to lymphoid organs, to lymphocytes, and the lymphocytes recognize this antigen and start to produce antibodies against the normal antigens inside the testes, and cause arthritis. So autoimmune arthritis, it's post-traumatic arthritis. So that's why my request to all of you, yeah, I said it to, I used to say that in histology, in embryology, and also in pathology. So uh, for boys especially, be safe with your testes. Yeah, no, so don't offend girls. Yeah, because girls are usually the first reaction to any offense is just kicking to your, you understand. Okay, let's continue. So what is the mechanisms of autoimmunity? We have general principles of autoimmunity mechanism. Autoimmunity arises from a combination of the inheritance of susceptibility genes, which may contribute to the breakdown of self-tolerance, and environmental triggers such as infections and tissue damage, which promote the activation of self-reactive lymphocytes. Okay, might be so. Why in autoimmune disorders are happen? For example, usually it's due to defective tolerance or regulation. We have so all these mechanisms we discussed about the tolerance, they are might be not regulated due to inherited diseases. And actually, in most of our autoimmune disorders, we have specific gene patterns. So we have specific genetic components and they are known as Mendelian inherited autoimmune disorders. Um, okay, in some details it will discuss later. Then abnormal display of self-antigens. Abnormalities may include increased expression and persistence of self-antigens that are normally cleared or structural changes in these antigens resulting from post-translational enzymatic modifications or from cellular stress or injury. Okay, it can produce new antigens, new epitopes that are not expressed normally, and the immune system may not be tolerant to these epitopes, and anti-self response may develop. So as example of this abnormal display of self-antigens, it's diabetes mellitus. So you know that in diabetes mellitus sugar level is increased, and self-antigens are present here, so they are usually glycoproteins. And when glucose level is too increased, so you know that we have a reaction of polymerization, not polymerization, polycondensation reaction is very typical for carbohydrates. And glucose can bind so by covalent bond to the glycoprotein, and this glycoprotein became different. This new antigen happens, yeah, and our own cells became antigenic for us. That's one of the best examples of this abnormal display of self-antigens. And then inflammation or an initially innate immune response. Innate immune response is a strong stimulus for the subsequent activation of lymphocytes and the generation of adaptive immunity responses. Microbes or cell injury may elicit local inflammatory reactions that may be triggers for subsequent autoimmunity. So that is okay. You understand the three processes which cause autoimmune. So it's defective tolerance, abnormal display of self antigens, and just inflammation or initial innate immune response. So now here you see that they might be combined. Yeah, all these three of them might be combined. For example, defective tolerance or regulation. So usually it's genetic factors. But they might be inactive in some conditions. But if we promote it with the inflammation or we promote it with abnormal display of self antigen, it causes a very severe autoimmunity disorder. Okay, let's talk about genes. Susceptibility genes are susceptibility genes, so they cause the failure of self-tolerance, and then these genes we discussed before, they start to figure this our own tissue antigens like antigens, and then immunity reactions respond on it. So this is a role of susceptibility genes. And then association of HLA alleles with disease. So HLA genes, alleles, among the genes known to be associated with autoimmunity, the greatest contribution is that of HLA genes. HLA genes, so you remember the MHC complexes. MHC complexes, they are controlled by HLA. They're also known as HLA, and HLA genes are present in our body. So this I don't ask you to remember it. So just remember that HLA genes are responsible. So just this. And we have non-MHC genes with autoimmune diseases, so which are causing it. So there's also no need to remember this by heart. So, but you remember that it's always the receptors to cytokines. It might be receptors to cytokines, so which defectly which cause a defection in suppression of immune response. Okay. So, for example, infections and other environmental factors. So, for example, we have some infections. We have some infections which cause the cross reaction in our body. It's caused by molecular mimicry. For example, streptococcus. So the best example is streptococcus, hematolytic streptococcus type B. So it's alpha hematolytic streptococcus type B. So it on the surface it possesses antigens which are mimic to our myocardium, or mimic to endocardium, so to endothelial cells, and that's why in molecular mimicry, so that's why our body start to fight against it, start to produce antibodies against it, so T cell reaction against it. But then you know that we diminish the offending agent, but after this we have antibodies, and also we have T cells which are able to kill our myocardium and endocardium, and that's cause the disease known as rheumatism. Rheumatism, not rheumatoid arthritis, but rheumatism. It's very common. Oh, okay. Also, another viruses like ABV, EBV, and HIV. They cause the polyclonal B cell activation, which may result in production of autoantibodies, like additional factor. Okay, what else we can see? Ultraviolet radiation can promote the autoimmune reaction. So, okay, let's skip it. So general features of autoimmune disease. Autoimmune disease tend to be chronic, sometimes with relapses and remissions, and the damage is often progressive. The clinical and pathological manifestations of an autoimmune disease are determined by the nature of the underlying immune response. So you see, we have different diseases, and let's now just count them at least. We have systemic lupus erythematosus. Systemic lupus erythematosus is promoted by specific autoantibodies. So it's a type 2 reaction. Yes, so autoantibodies, which are known as anti-nuclear antibodies. ANNAs. So ANNA. So and you can suggest, yeah. So if we have antibodies against nucleus, so we can kill every cell in our body. Yes, it is, and it is, and everywhere where these antibodies appeared, they can cause the systemic lupus erythematosus. So we have very large variety of this lupus erythematosus clinical manifestation, but we have hallmark of SLE in the production of autoantibodies, several of which antibodies to double-stranded DNA and so-called Smith antigen are virtually diagnostic. So we can detect it. We can detect it in pathological examination. So also ANNAs, yes. So this ANNA, so anti-nuclear antibodies. Against nuclear antigens can be grouped into 4 categories. They're DNA, histones, non-histone proteins, and nucleolar antigens. But any of them can damage our cells, and they are killed. So these clinical manifestations are very, very abundant. So this in clinic, you will discuss it in details. So acute cutaneous lupus, chronic cutaneous lupus, non-scarring alopecia, oral nasal ulcers, joint disease, cirrhosis, renal disorder, neurologic disorder, hemolytic anemia, leukopenia or lymphopenia, thrombocytopenia. So it depends on which type of antibodies are produced. Immunologically, we can see the anti-nuclear antibody, anti-dsDNA antibody, anti-SM antibody, then antiphospholipid antibody. This is actually in distinct disease. It's antiphospholipid syndrome and a low complement. So low decreasing of complement. So I remind you. So especially C3 level is decreased due to spend of this C3 during the complement activation in this area. Okay, and we have direct Coombs test. So by Coombs test, anti-red cell, we detect the anti-red cell antibody in the absence of clinically evident hemolytic anemia. Okay, that's so we have different syndromes. So lupus erythematosus, systemic sclerosis, Sjögren syndrome, autoimmune myositis, rheumatoid arthritis. Okay, actually, okay, let's make it your self-individual work. So actually, we have no time to discuss it. That's why I'll make it as self-individual work. This all the types of this autoimmune disorders, starting with SLE. So all these diseases. So SLE, systemic sclerosis, Sjögren syndrome, and autoimmune myositis. So next week, first class of so please announcement for everyone. Next week, every first class of this, I'll ask you about this disorders, autoimmune disorders. And you have to explain in detail. So what is a systemic lupus erythematosus to make a just make a PPT or any kind of other type of presentation, and I will evaluate it for mark. Then systemic sclerosis, Sjögren syndrome, autoimmune myositis, and rheumatoid arthritis. So in next practical class, I'll distribute between you these topics, and then everyone will be responsible for this disease. Okay, that's it about this. And yeah, one more disease, which we need to discuss. So it's systemic lupus erythematosus, Sjögren syndrome. So what is it's also systemic sclerosis. So you can check it out here. Then IgG4 related disease. Okay, and then rejection of tissue transplants. Yeah, so you know that. So this I cannot give you for self preparation. So tissue transplants. So please, and tissue transplants are self antigenic. Yeah, so when you know that after the tissue transplantation, we use a relative tissue transplantation with a very common antigens present on it, but it's a very rare condition when we can detect the when we can find the organ which is similar in all the factors of tissue factors with a absolutely similar MHC. So that's why usually MHC is attacked with our immunity reaction. So recognition of graft alloantigens by T and B lymphocytes. Graft alloantigens, they're exchange between individuals of the same species. They are called allografts, and grafts from the one species to another, still an experimental procedure called xenografts. Most clinical transplants are allografts, as discussions focus on this. So allografts means that it's so during transplantation, so some antigens appeared on in your body, and that's related to HLA or MHC. Yeah, and you see, and you remember that HLA, if HLA antigens are a little different, so it leads to activation of CD8 positive T cells and CD4 positive T cells. And CD8 positive T cells, they predictably can destroy directly cells, but CD4 positive T cells, they activize the autoimmune reaction against it. So, and you see, it promotes the type 4 T cell mediated autoinflammatory reaction. So this is example of rejection of tissue transplants. Okay, so that's enough to discuss it. So what else? What else? So what is actually we can how to avoid the tissue transplant reaction. So actually sometimes during the transplantation, if we don't want to facilitate the so we don't want to have this autoimmune reaction, we can just um, change the stem cell, change the hematopoietic stem cell. Yeah, we can replace the all our immunity cells with the bone marrow of other patient. So with other patient who is not antigenic to this, then we can transplant the bone marrow to this patient, and it doesn't create the immunity against our own antigens. Okay, it's this treatment is still there, so it's in clinical subjects. You will discuss it. Okay, immunodeficiency diseases are skipped, so we can discuss it now. So usually immunodeficiency diseases are related to genetic disorders, which cause which promote the hereditary causes of decreased immunity states. It's also mhm. It's also self-individual work. Should be self-individual work. Okay, I'll give you the list of self-individual work little later. And of course, secondary immune deficiencies, and one of them is a very common. One of them, you know, it's HIV. So human, so HIV. So you know what's HIV. So human immune deficiency virus, which causes depletion of CD4 positive T helper T cells. Okay, so that's HIV also. I think it will be self-individual work. So, but not in so details, because it's shown too detailized. And one more disease I want to say to you, it's amyloidosis. Amyloidosis. So 1 minute. Amyloidosis, yeah. So this is was about HIV infection. So that's most of you already know about it, but in more details we'll discuss it in practical class. But amyloidosis. is a condition associated with a number of inherited and inflammatory disorders in which extracellular deposits of fibrillar proteins are responsible for tissue damage and functional compromise. So just want to show you diagrams. In easy words, so we have different types of proteins. Okay, let's show it here. So we have, you remember that we discussed about misfolded proteins. Yeah, misfolded proteins during the apoptosis. So, but sometimes these misfolded proteins, they don't cause the apoptosis in some conditions, but these misfolded proteins are collected somewhere, and they form some of these misfolded proteins with some after some reaction, it causes the formation of fibrils. These fibrils are deposited inside the cells. Then it might be acquired mutations, for example, and that's the most common type. So when plasma cells, they start to produce immunoglobulin light chains without heavy chains, and these light chains, they are not killed. They are not active immunity cells, and they start to deposit inside the tissues, and this is known as AL protein. So it has happened due to acquired mutations of B lymphocytes when it produces only light chains of immunoglobulin. We have a chronic inflammation. In case of chronic inflammation, so macrophage activation causes the releasing of interleukin 1 and 6, and liver cells produce SAA protein. And it is also can be deposited in the same way as in light chain disease, in the immunoglobulin light chain disease, and AA protein, and also might be just due to mutation inside the protein synthesis. So it produces a mutant transthyretin, and it produces a specific ATTR protein. So that's all of them. They cause the same idea. So amyloidosis is a collection. Amyloidosis is a collection of these proteins inside tissues, and especially tissues are and where we recognize it the first in kidneys. And first of all, the deposition of this amyloid in kidney tissue, it causes a lesion in kidneys, and they can't provide their normal activity, and then normal filtration is not happening, and it causes chronic kidney disease and causes kidney failure later, and it's very hard. So unlike other diseases, it has manifestation when it's too late. So manifestation of the disease happens when it's too late to heal the kidney, because it goes very, very gradually. So with a long term, and it's also the same amyloidosis happened in the brain. So in the brain, so don't forget about the brain amyloidosis, and actually it gives another disease. This disease is known as Alzheimer's disease. So one of forms of Alzheimer's disease is amyloidosis of the brain. So it's a deposition of this light chains of usually it's light chains of antibodies which are deposited in the brain. Okay, this is you see, very tough topics. A lot of things I just skipped for you because no need to remember it. So please be ready for in the next class. Goodbye. If you have any questions, you can ask.